The overall goal of this laser microdissection procedure is to develop a robust and reproducible method for the isolation of a highly enriched trabecular meshwork from mouse eyes using laser capture microscopy for subsequent RNA isolation and downstream expression analysis.
This method can help answer key questions in the ophthalmology field, such as what are the important genes and signaling pathways that, when misregulated, can lead to ocular diseases. The main advantage of this technique is that high-quality RNA from the trabecular meshwork of mouse eyes can be reproducibly isolated for gene expression analysis.
Generally, individuals new to this method will struggle because laser microdissection is a powerful technique which involves various troubleshooting steps throughout the entire protocol.
Begin this procedure with optimal tissue collection for laser microdissection, as described in the text protocol. Spray RNase decontamination solution onto the brush, forceps, coupling jars, and mounting chuck. After wiping thoroughly, rinse the apparatus with nuclease-free water and dry. Wipe down the cryostat with 100% ethanol to avoid cross-contamination. Squeeze a small amount of OCT onto the mounting chuck. Then, remove the tissue block from the mold and carefully place the block on the mounting chuck. Once the tissue block on the mounting chuck is frozen solid, insert onto the specimen holder of the cryostat. Next, adjust the cryostat to cut 8-micron sections and carefully section through the OCT block to reach the tissue.